Hello, everyone. Good afternoon. It's good to see so many of you joining in. And I think in a minute or so, we'll have more people joining in. So today we are all here for an open house session on IIT Jodhpur Advanced Certification on Next Generation Omics Technologies and Applications for 2024-2025. This is a 6 months intensive program training designed to give you inputs and knowledge about next generation omics and technologies. And this is with this course and training, you get access to IIT Jodhpur faculty and industry experts. And more about this course will follow from our speakers who are going to be speaking in today's session. So I'll just introduce a brief about myself and all the speakers who are going to be talking today. So I'm Arushi. I'm the program manager for digital initiatives at India Bioscience. And today we are going to have four speakers with us: Debjani, Shri Gauri, Pankaj, Mohit, and then we'll have a Q&A session towards the end. So Debjani is the Assistant General Manager for Marketing at Premas Life Sciences, and she'll be talking about unlocking the human genome, how clinical genomics is advancing disease prognosis and everything else in and around next generation omics. So Debjani will be followed by Shri Gauri, who's from Omics Technologies, and she'll be introducing the course to you all, followed by Pankaj, who's the assistant professor and the director for the course, and he'll be talking about the curriculum of the course. Mohit, eventually, who's the CEO of Omics Logic and the program advisor for the course, will be talking about the career options post the course and how and where you can start with the registration process in detailed steps about the registration process. And post that, we'll have a Q&A session. So every speaker has 15 minutes of talking. So without further ado, I think I'll hand over to Debjani, and Debjani, you can start. I'll stop sharing.
Hi, everyone. Yeah, I hope I'm audible.
Yes, you're audible.
Okay. Thank you so much, Arushi, for the introduction. And I'm really excited to be speaking to all the registrants here. And it's definitely, like the topic of my presentation is unlocking genomics.
Debjani, I'm not able to hear you. I'm not sure if that's only with me. Uh, Your voice is getting cut. Is it only for me? Can you confirm?
Yes, I guess the
What
voice
I can
is.
do is, okay, what I can do is I can just turn off my video to make sure that the
Maybe.
bandwidth is, yeah. Is it better now?
I mean, for now, we can hear you.
Okay, great. So, uh, although my presentation is titled Unlocking Genomics and How Clinical Genomics is Transforming Disease Diagnostics, I would go a step further and say that clinical genomics is not just transforming disease diagnostics, but it's also transforming the entire way we are actually viewing personal wellness, right? Because at the moment, there's a lot of impetus on, preventive rather than curative, and transforming care is basically very, very indulgent of patient care. But not just that, it also is extremely focused on population well-being. So, across the various domains of across various domains of patient care, or essentially, let's say disease research, there are solutions in genomics and rather multiomics which can address very, very pertinent questions in oncology, reproductive health and pediatrics, cardiovascular diseases, chronic and metabolic diseases, transplant medicines, neurological diseases, and infectious diseases. So, across the entire panorama of various diseases and patient care through all the different disease domains, there are solutions which currently omics is trying to address. And if you just see the entire life cycle of an individual, then at every point of time, genomics has something to offer. Okay. Genomics has something to offer. Let's say right before conception, when you're actually in the stage of family planning, to in the process of conception, let's say a prenatal test, newborn screening or assessment. Once the individual is born, there are multiple layers or multiple stages at which disease screening is done. Early and accurate disease diagnostics, let's say therapy selection, whether or not a therapy is going to work. And once the therapy is administered, whether the disease is relapsing after remission. These are all parameters that can be studied effectively using omics. And not just that, like earlier, there was a thing about medicines for all. But right now, we are at a stage where we're looking at personalized healthcare. What does that mean? That one size does not fit all. And you basically go about doing individual omics to figure out what kind of a care will be delivered to the person, so that a person has an effective kind of a therapy. Not just that, even in terms of the family lineage or the family disease details, you're able to cater to the person's screening very, very effectively, so that the disease state does not come, or maybe it's delayed at some point of time. So especially like something that is really well established at the moment is next generation sequencing, which I'm sure all of you have a little taste of. So next generation sequencing especially offers a lot of flexibility, which is basically needed to characterize every other omics associated with disease research and patient care delivery. So whether it's proteomics, epigenomics, genomics, transcriptomics, all of these different kinds of omics. So when you talk about proteomics, obviously proteins are the main functional entities. So they go one step ahead of genomics. So when you actually talk about DNA to RNA to protein, this entire central dogma, every part of it has a certain omics associated with it, and which in turn can be well linked to the various parameters which are necessary to study to understand the various layers of disease physiology. And in terms of the scalability, so at the moment, we're at a very interesting vantage point where we have a bunch of these different instrumentation which basically are necessary to unravel the various layers of omics. And interestingly, no matter what layer you are studying, whether it's a small virus to different layers of the host physiology, there are different scales of these platforms available, which whether you want to study a small genome or you want to study a whole human genome, or whether there's this concept of One Health, wherein you're actually looking at everything which is present in the ecosystem, which would be insightful in the well-being of an individual. So whether it's a bacteria present in the environment to a plant, which is, which serves as, let's say, food for all of us, to animals and to human beings, from where, which is supposed to be the most superior species on the earth, all layers, and even within an individual, whether it's genomes or the coding part of the genome, which is the exome, or let's say a whole RNA sequencing, or let's say just simple gene expression of few selected genes, or whether it's a targeted panel, or if it's cell-free DNA, which is also called liquid biopsy. All of these parameters can be studied using the vast a plethora of sequencing systems available in the country, okay, in the world. So, regardless of the throughput and regardless of the scale of your organism, there is something that can be utilized to study the problem at hand. And in terms of the various parameters or various applications that these systems cater to, whether it's, let's say, oncology, where people can go from studying single genes in multiple individuals to multiple genes in a single individual to comprehensive genomic profiling, which is actually a compendium of, let's say, 500 to 1,000 genes, which people often study to get an entire picture of what's going on in the individual which has cancer, to, let's say, transfusion medicine, where HLA typing is one of the key things that people study to see if there's a good amount of match, whether it's a solid organ transplant or a bone marrow transfer. Whether it's a rare disorder, genetic disorder, wherein, whether it's single gene or multiple genes which are causing a diseased condition. It could be ataxia, it could be any kind of a muscular dystrophy, or a whole genome or an exome approach where none of the targeted panels of genes have yielded anything. So you actually go directly for the big haul, which is a whole genome study. Or in expectant mothers, where you actually want to study the cell-free DNA and figure out if there are any kind of chromosomal aneuploidies in the fetus at hand. Or simply what they say, studying the gut. As they say, the gut is the second, and in some cases, people are referring it to be the first brain of an individual. So when you actually wanting to study the gut, a 16S approach gives you a lot of information as to what are the species of microorganisms harboring in your gut. So all of these different parameters can be studied very, very elegantly, and they've been demonstrated to yield fantastic results through a body of evidence generated over the past decade or so. Of course, not everything is hunky-dory. So while you have all of these pros, the fact remains that in terms of, let's say, so the costs have come down. Let me first tell you. Like we used to have this whole thing that sequencing or genomics or any kind of omics is very expensive. It is still not the most inexpensive technology, but from the days of the first assembly of the first genome to today, the costs have come spearheading south, right? So, and that's a good news, because an exome which used to cost almost 1 lakh only about 5, 6 years back is now costing let's say, sub 10,000 rupees at the moment. So the sequencing costs have dropped, but still, there is scope to bring it down further. And also that, each test that you do is accompanied with a lot of data, which might be complex, or there might be incidental findings, or there might be confusing results, which has to be interpreted and also has to be communicated to the patients, right? And of course, there's limited availability of use case of genomic testing within the country, especially because clinician awareness is still very, very low. So this is what we are trying to change, and this is what we hope all of you collectively will be able to change once you're through with the course. And when you actually talk about, accelerating the access to clinical genomics, these three things have to come together. First of all, there has to be innovation. The technology is there, but if you don't have a question to answer, if you do not have a research which can make use of the technology, it is absolutely of no use. So it's important that you have these questions, that you have, to answer through the use of technology. And also, when there's a clinician which has an evidence of improved patient care, that has to be communicated in the fraternity so that more clinicians are made aware of the use case scenario of the technology, and they're able to utilize it in their line of work. And there has to be a massive education drive accompanied by, so clinical testing has to go hand in hand with research, right? So when you talk about innovation, there are two kinds of innovation. Innovation in research, but also innovation in the technologies which are being used. For instance, Premise represents this company called Illumina, and Illumina, over the 18 or 20 years of their existence, have constantly innovated to bring the technology down. To bring the costs associated with the technology down from several thousands of, dollars to about, let's say, $10 per gigabase or $5 per gigabase, or at scale in some of our highest platforms, it's about $3 per gigabase. So, there has to be a constant evolution of the technology, not just in terms of the features which are available on the systems, but also in the applications that they support. And the applications part are mostly looked into by different scientific groups, which actually have now, we've reached a stage where methylation or proteomics or transcriptomics, everything is studied through the use of next generation sequencing. So you don't need like 50 different technologies to study 50 different applications. You can do it all in one platform. Not just that, the current generation of sequencers have also, the current innovation and the sequences which have resulted because of that are also very mindful of the environmental challenges. So there is a constant effort in the technologies to make their solutions eco-friendly and environmentally sustainable. For instance, the latest sequencers which have been launched from Illumina's tables have all got room temperature shipment and storage. So imagine if you don't have to use dry ice to ship your, reagents. It's going to essentially cut off the dry ice. If it's going to essentially be less of a burden to the environmental sustainability. Not just that, there is also reduced plastic use. There's, a higher use of recyclable plastic, etc., etc. So environmentally also, they have to be sustainable to make sure that you're not burdening the environment as you're innovating. And if suppose similarly, through a constant innovation of the sequencing systems, if you're able to arrive at your solutions faster, so there's a constant effort in working on the background chemistry, where things have evolved to a stage where you can finish the entire sequencing run and arrive at the results in the course of one single day. So there are labs in the US which actually order a new, the doctors go into the wards, hospital wards. They see the sick babies, the sick newborn babies, which would probably benefit from whole genome sequencing. They would order a sequencing sequencing based test to be done in the morning. And this is, I'm talking about a whole genome sequencing test. By evening, when they are done with the lab rotation, when they are done with the ward rotation, they get the reports in their hand. So how much faster could, your results be that you're actually getting to see the entire blueprint of the genome in the span of one day, right? So like I said, all sorts of applications are, enabled on some of the latest platforms. And especially with the reduced turnaround times, with the increased accuracy, and with the environmentally sustainable reagents shipping at room temperatures, the plethora of applications which are addressed on the systems, the new systems are really shattering the glass ceiling. And like I said, as far as the applications are concerned, few of the things which basically enhance the access of patients to genomics is, A, you have to increase the diversity. So a lot of the data we still have in the repositories are still, they're white. They're not brown enough. So essentially, they're all mostly Caucasian. And the representation, let's say, of Indian data is, about 0.2%, despite the fact that we have a seventh of the world's population in our country. So you have to increase the diversity so that, like different kinds of data are included in the representation of the genomic data that's there in the databases. The second thing is you have to have more reporting based solutions so that genomes can be interpreted, because clinicians do not have time to actually go through these reports. ensure everyone has access. So it's not just enough to have genomic data, but if suppose those data is helping you make a decision about certain therapies, those therapies should be available to the people at large. So it's important to have everyone, it's important that everyone has access to life-saving treatments tailored to their individual genomics data. And of course, for under-resourced communities, you make provisions through CSR, through philanthropic funding, so that they have access to these data and treatments too. Especially for oncology, now the trends are like people used to actually study single gene. Like for instance, in non-small cell lung cancer, people used to study EGFR, NRAS, KRAS, etc. as single gene testing. So when one gene used to not work, people used to go to another gene and to another gene and to another gene. And this used to basically not just take a lot of time, but often people would just run out of tissues. So now with the newer paradigms of oncology testing coming up, like for instance, the comprehensive genomic profiling, which especially can lead a lot of interesting and very very insightful views to a person's reports. Like for instance, in a study which included 561 non-small cell lung cancer patients, people saw that about 27% of people who were tested had negative results from single gene testing. And out of that, about 46% who actually had these negative results from single gene testing had a positive result when the number of genes studied was increased from 1 to about 500. And because of that, people got their diagnosis. And even if it leads to an increase in the mean survival of the patients by a few years, those few years are so precious that it's all worth it to have, genomic testing done, right? Similarly, another application which is really catching up is non-invasive prenatal testing, where conventional screening like the double marker or triple marker tests in pregnant women, which is often a part of a lot of pregnant women don't even get these double marker, triple marker tests done, especially in the rural and semi-rural segments. But in urban segments where people do get these tests done, about 4 to about 10% of false positive rates are seen, which means, there's a unnecessary trauma that the expectant mother goes through. However, through genomics-based non-invasive prenatal testing, the sensitivity and accuracy can basically hit about 99.9%, and hence it's a much higher accurate test which can basically yield insightful results about any kind of aneuploidies which might be there in the fetus, right? And like I said before, last but not the least, it's very, very important that more people are educated about these solutions. So this was the thought behind developing a genomics education program. As I speak, the number of clinical tests that are being done in the country range from anywhere about 1.5 to about 2 million tests. Okay. And when you actually have so many tests being done in the country and so many of the molecular diagnostic labs adopting genomics or any kind of omics, it's important to have people who have the knowledge of the subject come and get employed in those labs and basically assume responsibilities of spreading this entire thing and taking it to the new heights and educating clinicians and building genomics literacy, etc. And that's basically the foundational reason why this course was started. So it would basically include building genomic literacy, practical hands-on workshop, which basically gives an immersive experience to the end users. And there would be a lot of interfaces of a collaboration between the academia and the industry. That's about it from my slides. I'd be happy to answer any questions.
Thank you, Deepjani. It was indeed a very interesting talk. And maybe you can stop sharing your slides, and Shri Gauri, you can then share your slides.
Okay, thank you so much. I'll start sharing my screen. All right, I hope my screen is visible to all and my audio is also working fine.
Yes, we can hear you and see you.
Okay, perfect. All right, so thank you so much, Debjini, for the wonderful talk. So in my part of the presentation, I would like to introduce the participants to the IIT Jodhpur Advanced Certification on NGS OMICS Technologies and Applications. So during the slides, you must have already understood the growing relevance of the field. So to address this gap between the growing demand for skilled professionals in this field, IIT Jodhpur, Premas Life Science, and Omens Logic have come together and designed this unique program on next generation omics technologies and application that is designed to equip participants with skills and knowledge necessary to design, execute, and analyze multi-omics experiments. And in this program, we cover a wide range of topics, including next generation sequencing, multi-omics technologies, computational methods, and statistical approaches. And I can see that a lot of the participants who are with us right now have already registered for the program and might already be familiar with the program features. But for those participants who are new to this program and would like to learn more about it, in this part of the session, I would like to briefly introduce. So the training is divided into 3 flexible options. That is the basic certificate program, the advanced certificate program, which is the objective for today's session. We will be covering a lot more about this program in this session. And finally, the postgraduate diploma, which is a combination of the basic certificate and the advanced certificate program. Now, coming to the advanced certificate program, this is a 6-month training program that is designed to cover the topics in depth and provide participants with hands-on experience in the field of multi-omics and next-generation sequencing technologies. And some of the unique highlights of the program include the classes that are led by industry and the academic faculties from IIT Jodhpur, Premas Life Science, and Omens Logic. The classes are held over weekend, and 2 of the unique features of the program include: one is the mini project, wherein the students get the opportunity to work on a mini project co-supervised by the IIT Jodhpur faculty and the industry partners. And apart from that, campus immersion is also one of the unique highlights. We often get the question from participants, do we get any hands-on practicals during the program? So this is the opportunity that students get. We will explore each of these as well in the upcoming slides. And finally, during the entire program, participants are also trained in several of the open source scientific tools and algorithms. And finally, towards the end of the program, you will be able to gain 15 credits and be able to join the IIT Jodhpur Alumni Network. Now, one of the frequently asked questions that we get is, if you are a working professional, can you join the program? So the answer is absolutely yes. The program is particularly designed for working professionals from pharma, sports, nutrition, researchers from academia and industry, clinicians and service providers, as well as PhD or master students in bioscience, bioengineering, and other fields as well. All right. Now, we have covered so far about the program. Now, this is the team behind the program. We have a diverse range of faculty from IIT Jodhpur, as well as industry experts from Premas Life Science and Omens Logic, coming from different expertise in wet lab as well as dry lab aspects as well. And here is a quick glimpse into the growing relevance of the NGS. Debjini has already covered a lot about the importance of clinical genomics in disease diagnosis. If you can see on the screen to the left, we'll be able to see the growing relevance of the market from the projected growth rate of this market from 2024 to 2030, and it has grown and is continuously growing for the next few years as well. And regional wise as well, and the application wise, you can see here on the right, the stats that have been put together. So the regional wise outlook is the growing importance in North America, Europe, Asia Pacific, Latin America, and so on. And application-wise, the job market is tend to be seen mostly towards genomics, molecular phylogenetics, metabolomics, and so on. These are some of the topics that we are covering in the program as well to ensure that the participants are staying up to date with the latest tools and technologies. Now, I just want to give a quick overview of the program curriculum, but we will be covering about this much more in depth in the later part by Dr. Pankaj, who will be explaining much more in detail about the compulsory modules that the participants are required to take, as well as the elective modules that they can opt for. Now, coming to the mini project. Now, this is one of the topics that the students are mostly interested. So what is the mini project opportunity all about? And what are the topics that we can work on? So mini project, under this opportunity, students get to collaborate and complete a mini project under the course supervision of IIT Jodhpur faculty, as well as the industry mentors, so that they are able to apply their learning, whatever they have learned throughout the program, into a practical research questions. And here on the screen, you can see a glimpse into some of the topics that students have previously worked on. And the topics, research questions, are diverse in range, from bacterial whole genome sequencing, meta-analysis of differential gene expression, predictive modeling, developing R packages, and transcriptomic profiling as well. So this is just a glimpse into some of the projects that might give you an inspiration to work on your own. Now, coming to the campus immersion. So campus immersion is a wonderful opportunity where you're able to visit the IIT Jodhpur campus and get trained in the wet lab aspects of the NGS OMICS Technologies. So here on the screen, you can see a glimpse into some of the practical hands-on that the previous students from the batch were able to work on. So here they are performing sample and library preparation for whole genome sequencing. And apart from the on-site practical hands-on experience that they get, it is also a wonderful opportunity where you can directly interact with the mentors or from the IIT Jodhpur and industry partners, and network and grow your network as well. All right, so that is about the campus immersion part. And this is a glimpse into the students sharing their achievement, the various skills that they have gained throughout the program, and so on. And recently we had the convocation held for the batch. So this is a quick photograph of all of the participants with their mentors taking photograph at the IIT Jodhpur campus during the convocation. And the objective of the program is not only to get the participants trained, but we also try to improve our program based on the feedback that we constantly monitor the learning assessment forms that the students are filling out. We try to update the curriculum with the latest technologies and so on. So this is a quick glimpse into what the students have provided us with feedback. And as you can see, the overall feedback has been positive, whether that is providing with learning resources, they have been finding it adequate, and almost 87.5 students have been finding it, recommending the program. And this is just an overview of the course rating. Some of the most popular courses amongst the students has been the sample QC and processing techniques for NGS analysis and multi-omics research applications. So these are also the courses that we are covering under the advanced certification part of the program. Students are mostly finding the practical and technology-focused content particularly valuable during the program. And this is another frequently asked questions that we often get. So what are some potential job opportunities that we can look for towards the end of the program? So there are two aspects to this, wet lab as well as the dry lab aspect. We'll be covering about the career opportunities towards the end as well. But just to give you a glimpse into the wet lab opportunities first, whether that is in academia, industry, or through internships, it includes in job roles such as becoming a principal investigator, or continuing your PhD for higher studies, or as research scientist, clinical lab scientist, and so on. And in terms of computation opportunities, you can look for opportunities such as bioinformatics scientist or bioinformatics engineer, healthcare data analyst, or bioinformatics programmer, and so on. Now, which are the domains that students can potentially look into for such opportunities? It could be either in pharmaceutical and biotechnology companies, it could be in research institutions or laboratories, healthcare organizations and hospitals, data analytics and bioinformatic companies, as well as in the genomic diagnostic laboratories as well. So these are some of the domains that these students can look into for their potential recruiters. And these are some of the feedback that we have received overall. And most of the students have been finding the hands-on practical especially helpful, where sessions are held where they are supposed to read through the research papers and present their findings towards the end of the session to the mentor. So this is just a quick overview into the program. And if you have any questions, you can definitely reach out to us on our contact number. Our team member will share in the chat box her WhatsApp number as well as our email ID. So if you have any questions related to the program, you can definitely reach out to us as well. But you'll be also able to address any of the questions that you might be having during the Q&A section as well. Now, I would like to pass on the stage to Dr. Pankaj, who will be explaining a lot more about the program curriculum in depth and help you understand the overall courses that are being covered. So over to you, sir.
Hi, a very good afternoon to all. Thank you, Sri Gauri, for such a nice explanation about the features of the program. I hope I am audible.
Yes, sir, we are able to hear you.
And can you see my slides?
Yes, sir, we can see the slide.
Okay, great. So most of the important things have already been covered. So I will just talk about straightforward about the program structure that we are following. So we have this program split it into certificates, and if you combine the certificates, you get the diploma. But for now, I think it's important to understand about the certificate 2 courses that we are offering in the upcoming semester. So basically, we will have to offer this 4 compulsory courses, which are having category C, which is a compulsory course, and the category E is an elective course. So you will have to take 4 compulsory courses, including the mini project, which was already discussed. And 1 additional elective course. I will show you a list of electives that are available for you to take up. So this entire certificate 2 will have 15 credits, and more details about the course content of each of these courses is available at IIT Jodhpur website. So you can reach on this link about the details of the course contents. Yes, so these are the elective courses that I was talking about in previous slides. So you can certificate 2, you can take up these options, you can pick up from these options as an elective. So you will have courses such as protocols for clinical next generation sequencing applications, or omics-based precision medicines, computational personal genomics, artificial intelligence in healthcare, AI ML based rational drug discovery, computational structural bioinformatics, applications of omics in plant sciences. And this is the credit structure. So all of these courses are three credits, and the 202 basically means that it has a laboratory component. So all of these electives, except the last one, they have the laboratory component as well. So I think this is already explained, like your journey. So you, for the time being, I believe that you will be, and all of you will be entering over here at this entry at certificate 2. So you will go through this 15 credits of learning for compulsory courses, 1 elective, and then you will exit with a certificate 2. Right. So I think this part might be also have been talked about in depth by our company partners. So yeah, I think this is more or less from my side. So I'm thankful to all the our course instructors, our mentors, and as well as our administrative supporters who are very helpful throughout the program to assist you in any kind of administrative issues. So yeah, thank you from my side, and I hope most of you will uh, join this program, and I look forward to meeting you all of you in the courses. Thank you. Dr.
Okay, thank you,
Mohit,
Pankaj.
are you
Yeah, I
there?
will
Yes.
take,
Okay.
yes, I'll take over from here. Um,
Mhm.
Thank you for explaining all of this. I think we would want to cover all of this in much detail that we have already gone through. So I'll give you a glimpses of how things are and how we are seeing this to work. This is the second batch for the course, and we are really excited to have already a lot of people who are going through the course right now, completing from semester one and now going into semester two. Along with that, we are really also excited to get a lot of you who have already registered for the program. And to those who have not registered, I think this is a great opportunity because this IIT Jodhpur Advanced Certification Program in Next Generation OMICS Technology and Application is a very unique program. I think unique in a sense that it has the component of both advanced to basic to advanced bioinformatics data analysis, where we are focusing on next generation sequencing data, machine learning, R and Python. Along with that, we are also focusing on how to generate data. So you would have a very clear understanding from data generation, experimental setup, to then data analysis, which is certainly a gap that has been addressed in this program. And because of that, there is a lot of opportunities that you become eligible for. So I'll be talking about all of that. But before that, let me introduce our team here. So we are a US-based and India-based bioinformatics and research company, and we have expertise from really prestigious and people with a lot of publications and patents throughout their career, including Dr. Alfred Tauber, Dr. Leonard Brodsky, Dr. Julia Penov, and myself, and also our CFO, Mr. Kito Musa. So we have been also collaborating throughout this journey, and I'll discuss about that, how this collaboration can benefit you also. So we have collaborations with top universities. We have published several research articles with some of the faculties from these renowned universities, and we have done training programs for students for from an undergraduate level to a PhD level with many of these collaborators. And we are still working with many of these collaborators for several of these things. For the program, our team in India will be completely available for the entire duration of the semester, as well as helping you throughout the immersion and project process, as well as finally getting to your degree that you are sought for. And after that, also assisting with you throughout your application process and throughout your career requirements, whether that is a job, whether that is a PhD or a postdoc or a master's or some higher education related opportunities that you're interested in. So to focus on what we are actually doing here. So in this course, there will be weekend classes. So on Saturdays and Sundays, you will have classes from morning, and then there will be breaks, and then there will be, you'll be provided with recordings as well. And along with that, you'll be having practical classes where you will have one-on-one discussion with the mentors, and these mentors are really world class, and they are one of the top researchers, and they have shown how all of this works and are expert in the field of different type of omics as well as in data generation. Along with that, we have the team from Premas that really adds a lot of value from the perspective of data generation, understanding the sequencing machines, and doing the experiments. So that's how I think this makes a lot of sense, and it has all the components that are actually there. To address one of the questions that has been asked in the chat, so the project part and the mini project part is uh, is what you seek. So what type of project you want to work on, that is the first question we ask you, and that becomes more and more clear as you go through the materials and you learn with the mentors. You become more inquisitive, you have more questions, so by the end of it you come up with a research question, and that research question might have a component of core bioinformatics. It can have a component of data generation. It can have it can have an application based things like you're developing a package. So the mentor is decided based on those needs. So what do you need? And we have experts for everything for these things, especially in bioinformatics. So obviously not everyone knows everything. Everyone has their own expertise, and that's what you leverage as a student based on the research interest that you have. So that one key feature, and then the campus immersion. So the campus immersion, we have talked about it, but to tell you that it is an immersive experience where you kind of do the towards the end, meet with everyone and discuss and clarify all your doubts, and it's a great experience. Talking about the material that you'll be provided with. So many of you might have concerns about like whether you will be able to do it on time, whether you will have all the resources or not. How would you learn if some of the basic concepts are missing, and it is bioinformatics, it could be challenging, all those things are there. So this program addresses all of those questions as well. I'll just show you the resource that we have put together for this course, and I think it includes everything that we have, and that is a great opportunity that is being provided especially for this course, this program. So you will have unlimited access to Omics Logic resource. That's one thing. What this unlimited resource actually provides, I'll stop sharing, and then I'll share my desktop, and let me know if you can share, if you can see the website.
Yes, we can see it, Mark.
All right, thank you. So as I was saying, we have curated all this over the last 8 years, and we've been updating it based on the latest technology. And one of the challenges is that the technology is rapidly evolving, and how do we keep learning and as well as stay up to date? So this is how this entire platform is being used at and is going to be useful. For genomics, there's a question about variant calling. So it's just not about variant calling, but for genomics, there's a process through which we will take you so that you learn the basic beginner concepts, and you have access to also all of this all the time, so that you can revise and based on your availability. That is another issue that many participants have because of 6 months course, you get busy at times, there's a marriage or there's something. So how do you manage that? So I'll give you a demonstration. So we have projects based experiences. This is all simplified so that anyone can start learning and get from different levels, from beginner to intermediate to advanced. So we have elaborated resources that are tutorial based with video recordings and also graphical display. Everything is available in GUI as well as in command prompt. So once you learn about all of these concepts and apply them, and use popular tools like Galaxy, and also from command line, and then you have option for R. So you learn all of this in one place very effectively. You don't have to go anywhere. You can use this, and then there is a mentor who is always there to support you learn all of this. So that is a great advantage that you will get through this. And this also combines with the research program. So you are getting access to all the resources that is there, all the courses, which means that over 40 courses, different type of tracks. I was just, we have transcriptomics detailed, we have single cell transcriptomics, we have multiomics that we will cover in this one. You can choose between metagenomics, genomic studies. So there's a lot of options that you can choose from, but there is also a way that you can easily learn all of this in very quick manner and discuss with a mentor throughout this program. So that is one of the benefit of this program. This also combines our research program. So this is a very unique program that is one of our featured programs where you get to work on research problems. And you can see here in this page itself, there are 200 research projects that are published. So these are done by students in collaboration with a mentor, an external mentor or an internal mentor. You can have multiple mentors who can guide you throughout a project. You can reach out to a external mentor and we can work with them as well. So there's a lot of option, and there is a lot of different areas that you can work on, like oncology, neuroscience, infectious diseases, and you will dig deeper into the data sets and find relevant information using machine learning, and all of that is possible. So that is about the research program and how it connects. Now let me go back to my slides. Just a second. All right, so can you, okay. Let me know if the slides are visible. I'm resharing again, I think. All right. I hope it is visible. So I was talking about the case study, and I talked about the open source scientific tools and algorithms that that are the latest tools that are included in it. And in the end, there is 15 credits for you to take. So what is that is happening is that you're a part of a pathway that we have designed that comes with, basic understanding, beginner level understanding, to a curriculum that you're going through this entire process, and then specialization that you're choosing based on your interest, and then converting into a project idea, and then showcasing it for different career purposes. So we have these curated bioinformatics pathways that we have created for you to take, and then you can work on multi-omics research projects, and using also wet lab expertise and coding expertise based on your interest, and go through and develop your own project, and again validate some of it within the possibility of campus immersion. This is for 10 days that you will be in the campus towards the end of the semester, and it's an immersive experience that students have been really very happy about, because it gives us a complete understanding, end-to-end understanding of what we are doing. And then in the end, you're a part of the IIT alumni, being an IITian. And then who can do this? Basically, we have explained it, but people who are in their bachelor's degree, they are not eligible. You need to have a bachelor's degree to apply for postgraduate diploma. So if you have a bachelor's degree, then you can go for it, and then you need to submit your documents that will be reviewed. Now coming to the program fee. So the duration, we have already discussed. The credit we have already discussed. The intensive nature of the program we have already discussed. The effective cost of the certificate 2 is 1.5 lakhs, which is around 25,000. Effective cost is around 25,000 per month. And for certificate 1, it is 13 credits, and hence the fees. So about this entire journey, I can simplify this so that it makes sense and you can understand that how you can go about it. So you have to submit your application, which is like by paying a fee of rupees 300, you start that process. You're a part of the cohort now. We review your application, you receive an offer letter, and then after that offer letter, so the last date for submission for your application is 8th December. After that, we will be reviewing your application and you will be receiving offer letters, and you can start your payment schemes from there. We have a team that helps you out with, installment options or loan options if you require such options for your journey. And then the classes will start on 3rd January onwards, and it will run till April. And then we have the exams and certification and the campus immersion before that, and the final exams, and then we have the certification towards the end. So by this entire process, you kind of complete the certificate course, and then if you want to get a PG diploma course, you have to earn 13 more credits that you can opt for opt in the next semester, and get a PG diploma degree. About the career options, so we have a lot of students who went from developing doing this course or doing a research course and then going for higher education. So all of these are funded programs, by the way. So if I say higher education, and if you're earning like $1,500 or £2,000 in your PhD, that is a good income. So that is what is one of the achievable insights that has been done, and it is also being possible because we recommend many of these students. So there is a recommendation letter, and then helping throughout the journey is also there. So we review CVs and make sure that the information is significant. What we want to do is make you eligible for a lot of opportunities, not to give you like a guarantee that we don't want to give, because there are a lot of opportunity and you should be able to choose from it. So we would give you assistance and we will make you eligible for all of this, and so that you can choose the best option for yourself. So PhD, postdoctoral studies, and job opportunities, all of those are options, and publications and collaborations are also options. And there is another entrepreneurship track, that is another aspect that many of you would be interested in. So starting a new startup for your own self, or within your group, is also possible when you are trying to develop using this mini project, and after discussion and reviewing and getting a feedback from industry experts, that what product you can develop from using wet lab technologies and adding data analysis, machine learning, and all of that. Huge opportunities. I mean, if you are serious about it, there is a lot you can do in this using this program. The last aspect is to be able to help you in this entire process. So in India, we are also absorbing students who are Premas, Omics Logic, My Biome, IIT Jodhpur. So along with that, we have a broad network. So AIMS Jodhpur, AIMS New Delhi, and all our collaborators. So we help in that process. So if there is a requirement, we would give preference to the IIT Jodhpur students first and give them the information so that they can apply. And that is how that is being done in multiple ways. We usually have a WhatsApp group with all of you, and we will share all of this key information with you so that you can apply and get to that opportunity. And also then there is a LinkedIn group, so where a broad range of job opportunities are there that you can apply to and do this completely. So this is the process. To summarize it, to complete your registration, please submit the fee and share your information. We will send the follow-up email that will have all the documents required, that will be reviewed by IT team, and then you will receive an offer letter, and then you can go about it and complete your fee payment, and also offer different type of fee payment options that are there. And you can start right away from 3rd January to be a part of this course. It is over the weekend, so it will not be like you're going to be busy.
तो मेरा होगा
Yes, we are not able to hear you, Dr. Mohit.
Yeah, even I'm not able to hear. I think there's some issue.
All right, it seems he got disconnected. In the meantime, I guess we can open the floor to Q&A. So if the participants have any questions, you can start posting them in the chat box. So we'll start with the questions that have been put in the chat box in the beginning. So one question, I guess, Dr. Pankaj, you might be able to answer this. By Lobna is, she wants to know, are we going to approach variant calling in tumor and normal cell in the program?
Sorry, is there a question for me, Srivar?
Yes, sir. You or Dr. Pankaj can take up this question.
Yes, we are doing this. So it is also there in Omics Logic Resources. We have a project on this, and we are currently working in a project like this also. So yes, I think that is being covered already.
All right, thank you, sir. The next question is by Anamika. How the mentors are assigned to the students? Can they choose them by themselves?
Yeah, they can choose based on their requirement, because every mentor has their own expertise, and what expertise they seek for the project could determine. Also, they have an option to work with an industry mentor, work on wet lab project, work on dry lab project, work with IT mentors, work with the mentors that you have shown in the course flyer. So yes,
Thank you, sir. Vikash, would you like to unmute yourself and ask any question?
there are a couple of more questions there. So are you doing single cell genomics? We are doing single cell. We have resources for single cell genomics, but we don't have dedicated sessions for single cell genomics, because there are already like key technologies that we are covering, and you can obviously, you'll be comfortable to and do single cell genomics as well, and we will be able to provide you with resources that can help you perform single cell genomics. That is for Aki. Now Ritvi has another question. How to go about it PG diploma course if one has not completed certificate one? So this has happened previously as well. So you can opt for certificate 2, and then in June you can do the certificate 1, so that you can get the 28 credits required for PG diploma. I think that's how you can get a PG diploma course. So the fee is for everyone, the fee is same, and there are many components, and the actual cost is very high because of several factors. So there will be faculties teaching you all the time, and then there is wet lab NGS experiments, which are very expensive. So fee for the course for everyone is same, whether that is faculty or student or working professional. So we have different options. I think you can reach out to Rajni and our team members. They can help you out.
Yes, sir. So I guess we have missed one question. It is by Anamika. If the projects require multiple mentors, is it possible to work with both of them?
Yes, I think that is possible.
Do we have any other questions? Lakshmi is asking, she didn't do the certificate 1 course, so will she be able to do the advanced course?
Yes, actually, if you start now, in December, you can browse through the resources that we have covered in the basic course, and we have all those resources. So if you opt for advanced course, you will also get the resources, all the resources on Omics Logic that has all the basic concepts that you can quickly go through. and be ready for the advanced course or the certificate 2 course. Actually, it is an independent course, so it will be something that you can learn from independently from the certificate 1 course. We have a lot of topics that have been foundation courses that are covered in course 1, wherein you learn more about the experimental design and what are the common pipelines and all of that. But in this course also, we will have a overview of what we have covered, and then give you the details of so that you are more comfortable and you've been able to do it. So that is also our responsibility to make you um, comfortable with it. But you also have to put efforts and go through these resources and give some time to it. So the 300 is the fee first for registration, and then you get an offer letter. Then you pay the rest of the fee, which is 1.5 lakhs, and you can choose from different options that we are offering for you to pay that much amount. But the effective cost is around 25,000 per month. This course will add a lot of value, and you will have. So to answer Lakshmi, you will learn key technologies, the technologies that are really being utilized by most of the biotechnology, pharmaceutical, and all these different companies. I wanted to share with you this link where I posting jobs all the time. And Shrigari, can you or anyone can share that link to our group, where we will see that there's so many job opportunities in India as well as abroad about these technologies. For PhD also, it's the same. So when you plan for PhD and you want to apply for a PhD, if you have these skill sets, PIs will definitely would want you to join their lab. So it actually adds a lot of value in that aspect. And for paper publication, we will definitely be able to guide you. All of us here have a lot of publications that we keep on publishing, and we have top mentors and genomic experts like Professor Mitali Mukherjee. You can check her profile, and we have a lot of IIT Jodhpur faculties with us. So I think they can tell you about how do you go for a publication.
Thank you so much, sir. Do we have any other questions?
All right. Thank you.
All right, sir. Thank you so much, everyone, for joining. Thank you, Prema's Life Science team and India Bioscience team for organizing and hosting the session as well. So we are sharing the contact details once again in the chat box. So if you have any questions, please feel free to reach out to us. We will also share with you the brochure and associated details over an email. So please do check your inbox as well. Thank you so much, everyone. We look forward to seeing you in the program. Bye.